Hello and welcome to this week's What Were They Thinking? Among this week's news items we have a vaccine for cocaine and theoretically a treatment for addiction, a study claiming a relationship between red meat consumption and type 2 diabetes, and scientists find a way that cells communicate leading to cancer. The timestamps for these and other news items can be found in the description box below. Our starting line this week is cocaine and the theoretical vaccine that could help with addiction. Although, to be clear, this is nowhere near regulatory approval yet, so we are a long way from seeing it available. The fundamental idea is that by generating an immunological response to the cocaine molecule, the body is able to prevent it from ever getting to the brain. If it can't get to the brain, you don't get the positive response associated with it, and therefore, you can't satisfy the addiction. This, in a manner of speaking, will break the cycle of reward and consumption. Although, ideally, that wouldn't be the problem in the first place, but, well, we live in reality, so no matter what we want, ideally, this is the next best option. There is also a second benefit to this that's not necessarily as obvious, let's say. That is, it prevents issues for those who are addicted and consuming cocaine, passing on the implications of this to developing fetuses, effectively a newborn addiction to cocaine, which is a concern for those who are pregnant. Well, possibly a concern for them. They're more likely worried about getting their hit, and the pregnancy is not even second, it's probably a lot further down the line of concerns. The challenge with implementing this vaccine is going to be twofold. The first is using it with those who are theoretically over their addiction, that is, that have been rehabilitated. This would greatly reduce the probability of relapse. The second group is the one that's going to be the bigger problem, that is those who are currently on or addicted to consuming cocaine. They're the ones that would most likely have more benefit from it, but also suffer considerably more from what is practically going cold turkey. That is, suddenly cutting off any and all consumption of cocaine through the use of the vaccine. There is also some biological concerns. The biological concern is rather more direct. Simply put, if somebody were to consume cocaine and initiate an immunological response, the response would be rather substantial, and that would mean that their body would undergo a lot of negative results. Effectively, systemic inflammation, most likely feeling absolutely crap and like they have influenza, but some of the nastiest strains out there. And depending on their health, it could theoretically be deadly. This is why the first group, those who are in rehabilitation or who are no longer addicted, would be the best candidates. Shifting over from news that's positive to news that's slightly less positive and more an example of distorted facts. The MSU economics professor Mark Skidmore had an article retracted relatively recently claiming that COVID vaccines killed 278,000 people, a figure that has received a great deal of criticism, to be polite. The fact that it was retracted should tell you a lot about how bad the article was. This is where a post from Science-Based Medicine turns even more interesting. Various individuals, but particularly Steve Kirsch, have claimed that the researcher has been exonerated, and that term is used very loosely by him. Largely because, well, various groups have somehow found that the retraction wasn't founded. The underlying retracted article is something we criticized before because it relied very heavily on the idea of surveys and unverified reports being translated to real-world statistics, something that you can't really do the way that it was conducted here. This leads to the article not only being criticized by us, but by many academics, and the academics had a much greater impact on it eventually being retracted for fundamentally that reason. That is, as it is described in part of the retraction report, There was no attempt to validate reported fatalities, and there are critical issues in the representatives of the study population and accuracy of data collected. Yeah, that's a pretty harsh removal notification. What leads to claims that the researcher was exonerated is what the Internal Review Board or Institutional Review Board had to say about the research conducted. And as they write it, after a 7-month ethics investigation, Michigan State University's Institutional Review Board cleared Dr. Skidmore of any wrongdoing, stating they did not find any non-compliance to their protocols within the study. Dr. Skidmore's COVID shot study did not involve clinical work, but rather involved an online survey that posed little risk to human respondents. Due to his study being a survey only, the IRB at the onset of research determined Dr. Skidmore's study to be exempt from those clinical protocols and was cleared to proceed. That's not the issue with this. The reason it was retracted is that the methodological approach taken was not appropriate for what they were trying to do. This isn't an ethics issue, this is a method issue. And not just that, but it could kindly be described as a deliberately inappropriate methodology. Something that was chosen purely because it could be distorted. The idea of torturing the data until it says what you want, just slightly more obscure. Frustratingly, as Science-Based Medicine points out, the publication was not only retracted, but now it's been republished. Now, the problem with the republication is not just the whole copyright issue, but it's very much similar to the Seralini affair. That is, a bad study with bad methods, and as a result, results or conclusions that cannot be substantiated was retracted and then republished under a new journal, but the actual underlying issues leading to not just the retraction, but being lambasted by every academic who was competent, haven't been fixed. This is like taking your car to the mechanic, having the mechanic fix it, but not actually resolve the underlying broken thing, and then taking it to another mechanic who does the same thing again. The car is still broken. All you've done is take it to 2 different places. Science-based medicine goes into much greater detail on the, let's call it generously, research, but the fundamental flaws and all the problems that go with this. Mostly that the method is just not appropriate for what they were trying to achieve, and the various misrepresentations of what's happened. But the biggest issue here seems to be, well, twofold. One, that Michigan State University's Institutional Review Board has failed in their due diligence and done a terrible job, which is no surprise given that it's likely a number of bureaucrats who have no idea about what they're trying to do. But the second is that it was republished, although somewhat thankfully, the journal, and again we're going to use that term very generously, that it was republished in is a known anti-vax, publication. That is, it is filled in the editorial board with known anti-vaxxers who have various issues, and the biggest examples of this is the fact that one of their editorial members is a lawyer, not a scientist, who can actually edit on this. Plus, they also have some deceased people, so unless ghosts are editors and reviewers now, it's unlikely that they're doing much and the journal is poorly maintained, which again speaks to its quality. Shifting from studies that are at least slightly more substantial, or at least better suited to being able to demonstrate a correlation and some sort of value, one study has found a relationship between red meat consumption and type 2 diabetes, which is interesting given that meat is relatively low in any kind of sugars, which would normally be the thing contributing to type 2 diabetes, but, well, let's go into the details and you'll understand why this might be overblown, to be kind. First, let's put the study results in some sort of context, and specifically what they claim to begin with. They describe a hazards ratio, that is, if you were to take a normal person and a person in this situation and compare them, how much greater or what ratio is there between the two? For those who ate all red meats, it was 1.62, so basically a 62% greater chance. For those who ate processed red meat, so think of things like salami for instance, there's a 1.51. For unprocessed red meat, for example steak, there's a 1.4 hazard ratio or a 40% greater chance of type 2 diabetes. As with just about all media reporting, they've managed to make a mess of this, and it's just been summarized as there's a 50% increased risk of type 2 diabetes with red meat consumption, and some have even gone so far as to say only consuming red meat twice a week can cause this. So yeah, they're really overstating what's been found. They're basically taking the highest possible value and attributing it to all consumption of red meat, which isn't really either a reasonable or accurate description of what was found. If you were to look at the lowest consumption, that is basically 2 servings of red meat a week, there was a small but certainly not powerful increase, and as a result it couldn't be said to be a certain finding. And this is important. There is more, let's say, robust research out there, largely looking at larger groups of people over longer term, and largely more consistent, that consuming red meat only a few times a week is not bad for your health and to a certain extent necessary for iron. Although iron can be sourced other ways, but that's a different issue entirely. The other concern with it is that with those larger studies about red meat consumption and things like type 2 diabetes, those larger studies have found that once you start aggregating the data, the demerits or risk associated with red meat consumption are generally overestimated due to methodological issues. So it would appear that this study has fallen into the same trap, which is a problem. When you continue to do the same research with these same flaws, you're not making new research, you're making more bad data. And not just that, but their methodological flaws were purely based on correlation. And that's because they had several data sets that they then looked at the variables within, and this is one reason they found a relationship between type 2 diabetes and red meat consumption. Remembering that observational studies only find correlation, and correlation does not equal causation. As a result, because of this, they really can't draw the conclusions they have as definitively as they seem to believe they have. As science-based medicine also points out, with a quote, "Of note, individuals who reported consuming red meat most frequently within each cohort were more likely to eat less fish, fruit, and more calories, weigh more, and engage in less physical activity." In other words, they were the least healthy group and the ones most likely to be in a situation where diabetes would occur even if they were not consuming red meat. Put slightly less charitably, there were a bunch of confounding variables that they did not adjust for, and because they did not adjust for these confounding variables, the results are meaningless. All the more so when much more extensive meta-analyses and systematic reviews have found that red meat consumption has no impact on glycemic control and insulin risk factors for type 2 diabetes. Or in far more direct terminology, eating red meat won't cause diabetes at this point in time. Shifting to better, or at least far more established research, we have it could be called a novel antibiotic, but it's novel in that it could be used. It's not novel in and of itself, as the antibiotic is about 80 years old. It is one of the last, let's call them golden age antibiotics. That is, up until roughly the 1940s when this drug came out, there was a period where antibiotics were being discovered left, right, and center. As a result, many of what we have now are either variations on, derivatives of, or exactly the same antibiotics from that time, with a few minor exceptions. As a result, at least arguably, many of those drugs were shelved and put aside as they weren't as effective or they had complications associated with their usage, and so they were abandoned for all intents and purposes, which is good in some ways. But the key here is that there are a number of drugs that have been put to a side that are now being taken out of storage and are usable, this being one of them. The reason why it's useful in some ways is that, well, all the patents that were associated with them have long since expired. That means anybody's able to look at them and start developing them in a, let's say, novel methodology. That is, take them, modify them, and make them work better, and at least somewhat compensate for those negative aspects. The drug, at least when it was discovered originally, was called streptothricin. The new name it's going under is, well, noroceo-thricin, which is really just rebranding of the original drug, but, well, Harvard University is rather fond of making things sound fancier than they need to be. For example, discriminating against Asians is positive discrimination, which, well, is just another way of saying discrimination. The benefit of Harvard, rather than just its racial discrimination policies, is that this drug is actually beneficial. The challenge with its original formulation was that it was very damaging to the kidneys, which isn't a novel problem. Some of the more aggressive drugs we have today that are intended as antibiotics can cause damage to the kidneys, so monitoring of patients on them is essential. What this does is forgo that aspect entirely, which is exactly what you want to see. The only limitation to this drug at present is that we don't actually know how it's working the way it's working. We do know that it is effective at killing a number of medication-resistant gram-negative bacteria, which is pretty much the group that you're most concerned about. Surprisingly, gram-positive bacteria remain relatively vulnerable to most drugs today, and that's because their cell wall, the peptidoglycan wall within them, is vulnerable. Gram-negative bacteria, since they don't really have the same peptidoglycan wall, aren't as vulnerable to drugs that we have now, which is why they are the majority of these drug-resistant bacteria. And having yet another tool to take care of them is good. In other cellular news, and still focusing on medicine, but this time shifting to tumors and cancer. As a general rule, most tumors and cancer are caused by cells losing control of their ability to replicate, or more accurately, to stop replicating. When cells continue to divide without control, you have a tumor. When cells continue to divide without control and lose their function or no longer function correctly, you have cancer. In very simplified terms. But it often starts from one or two cells mutating this way, and that's how you get what's happening. The new research is a little more disconcerting in that it indicates there may be a way for cells to communicate cancer from one to the other, meaning that a cancerous cell can now turn a non-cancerous cell into something pathogenic. A rather disconcerting notion. The rather simplified version of these findings is that when cells divide, as cancer cells do quickly do, they leave behind a small, what you could basically describe as a vesicle or similar. This small vesicle is full of remnants of the process of cell division, and most importantly, it has genetic instructions for cells to divide. Now, research to date has mostly believed this to be something that is destroyed once the cells finish dividing, which is exactly what you want. But the research now has indicated that in some cases it may not be destroyed and instead may be picked up by other cells. The problem the research has identified with these cells that pick up these mid-body remnants is that those that are most likely to do so, and those that, for lack of a better way of phrasing this, consume most of these mid-body remains, tend to be the ones that turn into cancer and proliferate most readily. This means that the more the cancer cells divide, the more mid-body remnants are left, the more the cells divide again, creating a positive feedback loop. Further to the issue of the cancer cells basically being given a stimulus to multiply more readily, the RNA within these mid-body remnants also had an effect on gene regulation, and mostly it's focusing on both cellular machinery and oncogenes. As a result, it more readily shifts the cells around the cancer to favor becoming cancerous. Of course, exactly how they do this, what's going on and so on isn't well clear yet, but its discovery of this small, seemingly innocuous and often overlooked structure that could be contributing extensively to the issue of cancer, and may explain why some cancers are not responsive in the same way as others, or why some people are more vulnerable to cancer. The final news we have for you this week is about an alternative source of cannabis compounds. Strictly speaking, it wouldn't be appropriate to call them cannabis compounds in this case because, well, it's a different plant, not just that it's, well, no longer cannabis. Cannabidiol, or CBD, was found in a relatively common Brazilian plant, and as a result, there is the capacity to produce CBD via means other than just, well, having to grow the cannabis plant itself, something that could be useful if you are a consumer of CBD oil, for instance. The plant that makes it is called the Trima marcantha bloom. It's a small shrub, and it grows across most of South America. More usefully, it's considered a weed, which means it's likely to be very, let's say, resilient to poor soil conditions, meaning it could be more readily farmed than the various kinds of hemp or similar from which you would source CBD. The other advantage to using this weed is that, well, unlike the cannabis plant itself, it's fundamentally legal across all jurisdictions, assuming import and other regulations could be met, whereas growing cannabis, not so much, and in most places it's not legal. This means that you could more readily grow a crop purely dedicated to production of CBD without having to worry about any regulatory hurdles. The only caveat at this point in time is that, well, they don't specify, the sources we've read, just how much CBD could be extracted from the plant, and that could be a significant caveat to their research and any findings. That is, without knowing how much CBD can be produced, it could be a inefficient, ineffective, and therefore unsuitable source. Hopefully the 500,000 real, or roughly $100,000 grant from the Brazilian government will have a better idea, although it does say that will take 5 years to complete, a wait almost as long as those who are waiting for their father to return from going out to buy cigarettes and milk. That's all the news we have for you this week. Thank you for watching. If you found it interesting, consider liking, sharing, and subscribing. Please do post any comments, questions, or suggestions you have below.